New genome editing method just dropped, and it's very cool. Let me explain. For a long time, scientists have wanted to figure out how to harness retrotransposons for genome editing. Retrotransposons are jumping genes, big stretches of DNA that encode the machinery to copy and paste themselves into new spots in the genome. And fun fact, over 40% of your genome is made of them. Because they're naturally very good at putting large sequences into particular genomic sites, they'd be an amazing tool for genome editing, if we could control them. But it's been hard to make a retrotransposon efficiently insert a user-defined sequence into a user-defined location, until now, with STICHR. How did they pull it off? One key factor was finding a good starting point for optimization. To find a likely to be successful retrotransposon to tinker with, they searched through thousands of genomes, looking for elements that looked like they could have reprogrammable insertion sites. They chose one that could accept custom genetic cargo and optimized from there. Another critical feature was bringing Cas9 into the mix. Retrotransposons have to cut the DNA to insert themselves, but they can cut kind of haphazardly. Cas9 is great at finding very specific genomic spots, so they fuse a Cas9 variant to the retrotransposon to help guide it, and thus boost precision and efficiency. And finally, customizing the cargo was actually pretty straightforward. They swapped in their cargo for the native transposon sequence, adding some flanks to match where in the genome they wanted it to go, then optimize these little overhang regions that help facilitate the insertion process. So in the end, they have a system that's able to do targeted delivery of hundreds of base pairs with efficiencies up to 40%, but more often efficiencies are in the 1 to 10% range. Still, that's a really solid starting point for future work to expand on, and that'll be exciting to watch because this sort of system could be a big deal for therapeutic purposes, because it's a one-step system that can scarlessly rewrite chunks of sequence, perfect for fixing diseases caused by diverse mutations across a gene. So stay tuned for future updates on further hacking the retrotransposon.